There are 3 steps to attachment and entry. The HIV protein GP120 attaches to CD4 molecule on the host cell surface. The ENV gene is the viral gene that codes for GP120. Co-receptor binding. Conformation change of GP120 occurs, which changes the shape of the protein. The conformation change allows GP120 to bind the co-receptors CCR5 or CXCR4. Fusion. There is another conformation change which activates GP41, which is also coded by the ENV gene, which initiates fusion. GP41 unfolds and folds back on itself like a slinky, initiating the fusion of the 2 membranes together. Once inside the cell, HIV makes its genome accessible by releasing a viral capsid, which contains 2 single-stranded DNA genomes and 3 enzymes necessary for replication. These enzymes are reverse transcriptase, integrase, and protease, which are all coded by the POL viral gene. Reverse transcription copies viral RNA into a double-stranded DNA helix that the body will recognize. Reverse transcriptase makes an RNA-DNA helix hybrid of the viral RNA. Then reverse transcriptase digests the original RNA and synthesizes the other strand of DNA. This is the step where mutation occurs every time.
After the provirus is created, it is spliced into the host cell's DNA. Integrase chops off the ends of the proviral DNA, making sticky ends so that it can be integrated into the host cell's DNA, and leads the provirus into the nucleus. Once inside, integrase latches onto the host DNA, cuts the host chromosome, and inserts the virus. Then, if the virus is active, HIV takes control of the cell and utilizes the cell enzyme to make copies of its RNA from integrated viral DNA. Some of the copies serve as the genome for new viral proteins. Others become the mRNA that code for new proteins. When RNA goes through translation to make new viral proteins, it makes large infected protein molecules which are cut by the enzyme protease into smaller, more mature proteins that each have a different function. On the original viral DNA, the GAG gene makes 4 structural proteins, and the POL gene codes for all enzymes. The core proteins and the copies of viral RNA are then packaged in a structural shell built by GAG proteins. New envelope proteins are then inserted into the membrane of the host cell. The shells move towards the regions of the cell surface where GP120 and GP41 are embedded into the outside of the membrane. An immature HIV bud then breaks off the host cell's membrane into the plasma, where after developing the viral capsid becomes a mature HIV virus able to infect other cells.